For one to understand muscle contraction, we need to combine our understanding of the microscopic anatomy of a muscle cell with the appearance of the muscle fiber under a microscope. Recall that skeletal muscle appears striated or banded under the light microscope. As early anatomists and physiologists began to reveal this structure with the advancement in the technology of microscopes, imagine their curiosity and the questions that they may have asked themselves. One physiologist is a man by the name of A.F. Huxley. Huxley is a physiologist and biophysicist who won a Nobel Prize for the discovery of the propagation of action potentials of neurons, something that we're going to learn about in the next chapter. Back in the 1950s, Huxley observed that during muscle contraction, the length of the A band remained the same while the I band shortened. The popular theory of the day was that the contractile proteins, mainly myosin, was what actually shortened, producing the contraction. Huxley proposed the sliding filament theory as opposed to a shortening filament, and this proved to be correct. Before we study muscle contraction, let's describe the bands of the striations that are seen under the microscope and explain why they appear as they do. Looking at a two-dimensional drawing of one sarcomere, we see the ends of the sarcomere at the Z-discs on either side. The actin filaments are attached to the Z-discs, and the myosin filament sits between the actin filaments. The area where myosin and actin overlap will give us the darkest band under the microscope. This is the A band. And the area where only myosin exists within the central portions of the myosin molecule is the H zone. Within the H zone is the M line where the thick filaments are connected together. The area of the I band is where the actin filaments run to the Z discs. Essentially, one I band is the actin filaments from two sarcomeres that are attached at the same Z disc. Let's take a look at this in the actual specimen now. Before we watch a couple of animations of muscle contraction from your textbook, pause this video and review the microstructure of a muscle fiber, including a review of the structure of the myofilaments, the sarcoplasmic reticulum, and the triad.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers. The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross-bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.